Hello and welcome students. Today we will discuss about fatty acid biosynthesis. To start with an introduction, we have humans and other mammals store energy in the form of lipid, but energy is consumed mostly in the form of carbohydrate. Therefore, these organisms must have a mechanism for the conversion of carbohydrate into fat. And this process is known as fatty acid synthesis. Fatty acid synthesis are a class of compounds containing a long hydrophobic hydrocarbon chain and a terminal carboxylate group. Fatty acid synthesis in animals occurs in cytosol of liver, kidneys, brain, lungs, adipose tissue, and mammary gland. And in case of plant cells, it occurs in chloroplastoma. This process involves stepwise addition of 2 carbon units, and this carbon unit is supplied by the acetyl-CoA, which is derived from oxidation of glucose. Now, let's come to the fatty acid biosynthesis. Fatty acid biosynthesis requires a sufficient concentration of NADPH and acetyl-CoA. NADPH is generated by the pentose phosphate pathway, and with the help of maleic enzyme, malate oxidizes into pyruvate and carbon dioxide. Acetyl-CoA, which is the building block of fatty acids, is needed to transport into cytosol from mitochondria. Once acetyl-CoA is transported into cytosol, a series of reaction steps occur leading to the formation of fatty acid. First step is transport of acetyl-CoA. Since fatty acid synthesis takes place in the cytosol, the acetyl-CoA produced from pyruvate has to be transported out of the mitochondria. However, the inner mitochondrial membrane is not permeable to this compound. So, it requires a special subtle mechanism called acetyl-CoA subtle system. It is a subtle system in which the acetyl-CoA is first combined with oxaloacetic acid in the presence of enzyme citrate synthase to form citrate, which can readily crosses the mitochondrial membrane. In the cytosol, citrate is converted into acetyl-CoA and oxaloacetic acid, which is catalyzed by citrate lyase with consumption of 1 ATP molecule and coenzyme A as cofactors. Then, the oxaloacetic acid formed is again reduced into malate by NADH-dependent cytosolic malate dehydrogenase enzyme. Malate is again converted into pyruvate in NADP plus dependent reaction, which provides NADPH that is required for fatty acid synthesis. To complete the circuit, pyruvate, which is generated in the reactions, again re-enters into mitochondria. The second step is the formation of malonyl-CoA. Acetyl-CoA in the cytosol is now converted into malonyl-CoA in presence of the enzyme acetyl-CoA carboxylase. In this reaction, bicarbonate HCO3 acts as a carbon dioxide donor and ATP as energy source for the carboxylation. The acetyl-CoA carboxylase ACC enzyme is a multimeric protein. It contains biotin, biotin carboxyl carrier protein, transcarbozylase, and regulatory site. Conversion of acetyl-CoA and malonyl-CoA occurs in 2 steps. In step 1, biotin is first carboxylated in the presence of ATP, and in second step, carboxyl group is transferred to acetyl-CoA to form malonyl-CoA. This is the first committed step of fatty acid biosynthesis. So, acetyl-CoA carboxylase ACC is allosterically regulated. Next, in the step 3, is the activation of acetyl-CoA and malonyl-CoA. Acetyl-CoA and malonyl-CoA are activated by transferring to a small protein called acyl carrier protein (ACP) containing phosphopentane molecule with sulfhydryl group, catalyzed by the enzyme acetyltransferase and malonyl transferase respectively. This acyl carrier protein is a single polypeptide chain in prokaryotes, whereas in eukaryotes it is covalently bound to a multifunctional polypeptide chain known as fatty acid synthase. Characteristic of fatty acid synthase. Fatty acid synthase is a multi-enzyme complex in a dimer form consisting of two monomers. They catalyze all the reactions in the synthesis process. Each monomer contains an acyl carrier protein ACP, 7 different enzymes and 2 free sulfhydryl groups. It is active only in dimer form and all the components have different functions. In the step 4, it is the synthesis of fatty acids. After the activation of acetyl-CoA and malonyl-CoA in step 3, following reaction occur in sequence which is known as elongation step. So, elongation steps include 4 enzymatic reactions. First step is the condensation step. Here, an acetyl ACP and malonyl ACP are condensed to form acetoacetyl ACP with the release of free ACP and carbon dioxide. This reaction is catalyzed by acyl malonyl ACP condensing enzyme called beta-ketoacyl ACP synthase. Second step is the reduction step. Here, acetoacetyl ACP is reduced to form D3-hydroxybutyryl ACP using NADPH as reductant, and this is catalyzed by beta-ketoacyl ACP reductase. Third step is the dehydration step. This is the step in which D3-hydroxybutyryl ACP is dehydrated by removing water molecules to produce crotonyl ACP in presence of 3-hydroxyacyl ACP dehydratase enzyme. The last step is reduction step. The crotonyl ACP is again reduced by a second NADPH molecule to give butyryl ACP, which is catalyzed by enoyl ACP reductase. These 4 steps is the first round of elongation that produces 4 carbon molecule butyryl ACP. Now, the steps are repeated in a cycle, with malonyl ACP adding 2 carbon units in each cycle to Langdon acyl ACP chain. This continues until the 16 carbon palmitoyl ACP is formed after 7 rounds of cycle. Once palmitoyl ACP is formed, fatty acid synthase enzyme cannot elongate further. Therefore, palmitoyl ACP is hydrolyzed by a thioesterase giving palmitate and ACP as the final product. For each of the 7 rounds of fatty acid elongation, 1 ATP is used in synthesis of malonyl-CoA and 2 NADPH are used. Palmitate is the primary product of fatty acid synthase. Fatty acid with shorter chain can be generated by releasing the fatty acid before reaching 16 carbons of length, whereas fatty acids longer than 16 carbon atoms require special elongation reactions which occur either in the mitochondria or at the surface of the smooth endoplasmic reticulum. The elongation reaction that occurs in the endoplasmic reticulum is similar to that of fatty acid synthesis. Now let's come to the desaturation or unsaturation of fatty acids. Since fatty acids are of 2 types in nature, that is, saturated fatty acid having single bond an unsaturated fatty acid having double bond. So, additional reactions of adding double bond is required for the production of unsaturated fatty acids. Both prokaryotes and eukaryotes are capable of introducing double bond in a newly synthesized fatty acid. Palmitate and stearate serve as precursors of the two most common monounsaturated fatty acids of animal tissues. Palmitoleate having 16 carbon and double bond at carbon number 9, and oleate having 18 carbon and double bond at carbon number 9. The double bond is introduced into the fatty acid chain by an oxidative reaction catalyzed by fatty acyl-CoA desaturase. This enzyme is a mixed function oxidase enzyme. It consists of non-heme iron atom which help in desaturating the chain. The path of electron flow includes a cytochrome B5 and a flavoprotein cytochrome B5 reductase, where fatty acid and NADH or NADPH undergoes two electron oxidation reactions simultaneously. In plants' chloroplastoma, oleate is formed by a sterile ACP desaturase using reduced ferredoxin as the electron donor. But in mammals, the enzymes required to insert double bonds at carbon atoms beyond C9 in the fatty acid chain is absent. Thus, they cannot synthesize linoleate having double bond at carbon number 9 and 12 and linolenate having double bond at carbon number 9, 12 and 15. Therefore, in mammals, linoleate and linolenate are called essential fatty acids since they have to be supplied in the diet. These two unsaturated fatty acids are also the starting points for the synthesis of other unsaturated fatty acids such as arachidonate having 20 carbons and 4 double bonds. Arachidonate is known to be the precursor of several biologically important molecules, including the prostaglandins, prostacyclins, thromboxans, and leukotrienes. Now, let's come to the regulation of fatty acid synthesis. Regulation of fatty acid synthesis can be discussed under 3 levels. First one, nutritional regulation. In many animals, the excess carbohydrate in the body is stored as fat to provide energy at the time of need. In this process, when the concentration of glucose and intermediates such as pyruvate, lactate, and acetyl-CoA is in surplus, it is converted into fat, assisting the anabolic phase of this feeding cycle. The nutritional state of the organism is the main factor regulating the rate of synthesis. Thus, the rate is high in the well-fed animal whose diet contains a high portion of carbohydrate. It is depressed under condition of restricted caloric intake on a fat diet or when there is a deficiency of insulin, as in diabetes mellitus. Fatty acid synthesis is increased when sucrose is fed instead of glucose because fructose bypasses the phosphofructokinase, which is the control point in glycolysis. So, second is the enzyme allosteric regulation. Here, acetyl-CoA carboxylase plays a main role. It exists in two forms, an inactive form and active polymer form. When citrate binds acetyl-CoA carboxylase, it converts into active form from inactive form by phosphorylation, thereby activating the fatty acid synthesis so that the excess acetyl-CoA is stored as fatty acid residues within triacylglycerol in adipose tissue. But when there are high levels of palmitoyl-CoA, which is abundant when there is an excess of fatty acids, acetyl-CoA carboxylase reduces its activity and stop the further synthesis of fatty acids. Third is the hormonal regulation. Glucagon hormone is responsible for blocking the fatty acid synthesis by increasing the phosphorylation process, which is carried out in the presence of cyclic AMP, since active acetyl-CoA carboxylase is converted into inactive form. Next hormone is insulin. Insulin stimulates fatty acid synthesis by several other mechanisms as well as by increasing acetyl-CoA carboxylase activity. It increases the transport of glucose into the cell of adipose tissue, thereby increasing the availability of both pyruvate for fatty acid synthesis and glycerol 3-phosphate for esterification of the newly formed fatty acids. Now let's see what are the metabolic fate of fatty acid. Fatty acids are oxidized to acetyl-CoA for energy production in the form of NADH. Fatty acids can be converted to ketone bodies. It can be used as fuel in extrahepatic tissues. Palmitoyl-CoA is a precursor of mono and polyunsaturated fatty acids. Fatty acids are used for the biosynthesis of bioactive molecules such as arachidonic acid and eicosanoids. Cholesterol, steroids, and steroid hormones are all derived from fatty acids. Excess fatty acids are incorporated into triacyl glycerols for the storage of metabolic energy in adipose tissue. They are also incorporated into the phospholipid forming structural components of membranes. So let's see its biomedical importance. Any disorder in the pathway of fatty acid metabolism could lead to various diseases like diabetes, obesity, and metabolic syndromes. Deficiency of essential fatty acid in humans include skin lesion and impairment of lipid transport. They are also required for prostaglandin, thromboxane, leukotriene, and lipoxin formation, which are found in the structural lipids of the cell. Abnormality in the metabolism of essential fatty acids cause several diseases like cystic fibrosis, acrodermatitis, enteropathica, multisystem neuronal degeneration, Crohn's disease, cirrhosis and alcoholism, and Ray's syndrome. Drugs like hydroxycitrate target the citrate lyase that block the enzyme to act on its natural substrate citrate. As a result, availability of acetyl-CoA for fatty acid synthesis is impaired. Diets with a high polyunsaturated and saturated fatty acid ratio also reduce serum cholesterol levels and are considered to be beneficial in terms of the risks of development of coronary heart disease. Fatty acid synthesis is a process involving multiple enzymatic activities. Palmitate, which is the primary fatty acid, are normally produced by the cytosolic fatty acid synthase complex. Other fatty acids are synthesized by addition of 2 carbon units by elongation reaction. Like any other metabolic pathway, fatty acid synthesis can be controlled by enzyme allosteric, hormonal, and nutritional regulation. Any disorder in the synthesis pathway or deficiency of essential fatty acids leads to several diseases in humans. So, the study of related enzymes such as acetyl-CoA carboxylase, citrate lyase, fatty acid synthase have clinical possibilities for the production of novel antibiotics, drugs, and the development of new therapies.